Cloning is a process used to create similar population of genetically identical individuals. The most common cloning method is DNA cloning. It used to create an exact copy of an organism by using the complete genetic material of a regular somatic cell. The components that needed in a cloning process are desired DNA fragment, vector, restriction enzyme, and DNA ligase. Vector is a small piece of DNA taken from a plasmid or a virus or the cell of a higher organism that can be stably maintained in an organism, and into which a foreign DNA fragment can be inserted for cloning purposes. There are a large number of cloning vectors are available. The plasmid is the standard cloning vectors and the most commonly used. A restriction enzyme is an enzyme that cuts DNA at the specific recognition nucleotide sequences known as restriction sites. These enzymes are found in bacteria. There are 4 types of enzymes based on their composition and enzyme cofactor requirement, the nature of their target sequence, and the position of their DNA cleavage site relative to the target sequence. DNA ligase is an enzyme that facilitates the joining of DNA strands together by catalyzing the formation of a phosphodiester bond. Below are the steps for cloning process. First, isolate the DNA from the organism that contains desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. The restriction enzyme that we use is EcoR1. It cuts in specific sequences in the DNA, generating fragments with sticky ends. Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme.
Now we proceed to the next step, that is insertion of recombinant DNA plasmid. A single DNA molecule that combines from 2 different sources of DNA is called as recombinant DNA plasmid. Recombinant DNA plasmid is introduced into living bacterial cells such as E. coli that are competent to accept the plasmid DNA. The next step is to make the host bacteria is competent. There are 4 steps to make the bacteria take up DNA from surrounding medium. Firstly, incubate the cell with 100 millimolarity calcium chloride solution at 4 degrees Celsius for 30 minutes. After that, centrifuge the cell for a few seconds. Then, resuspend the bacterial pellet in 75 millimolarity calcium chloride solution that contain 15% of glycerol. Lastly, freeze 0.2 ml of cell sample in the freezer. The 8th step is transformation of bacteria. Transformation of bacteria and collection of a cell containing the recombinant plasmid is by thawing the sample on ice. It also can transform by adding recombinant DNA on the cell sample. Besides that, the cell can transform when incubating the suspension at 42 degrees Celsius for 90 seconds. Lastly, spreading suspension on the agar plate containing selective antibiotic can make transformation of the bacteria. The last step is each cell containing a recombinant plasmid that produce a colony consisting of millions of progeny bacteria with the same recombinant plasmid. Thank you.
Created using Powtoon.